Hi, myself I am R.Bhushan. Welcome to ASG अभिनव स्टडी ग्रुप साइंस एंड टेक के इस प्रोग्राम में मैं आपको बताऊंगा क्रायोबायोलॉजी के बारे में. क्रायोबायोलॉजी, व्हाट इज क्रायोबायोलॉजी एंड इट्स फंक्शंस and different dimensions of cryobiology. Cryobiology deals with effects of freezing and low temperature on living organism. Cryobiology deals with effect of freezing and low temperature on living organism. In recent times, focus of cryobiology technique has been expanded to non-living things. Temperature is a measurement of the internal energy in a physical system. It is this internal energy is fluid system that allows molecules in fluids to tumble, twist, disassociate from one another, move from place to place in the fluid and chemically react with other molecules. As the temperature is reduced, lesser energy exists in the system to drive these kinds of molecular motion. In many systems, such as pure water, temperature reduction below a certain point result in an abrupt reorganization of fluid medium into an organized solid lattice known as crystal. Organized solid lattice known as crystal. Organized solid lattice known as crystal. In other system, this does not happen. Instead, temperature reduction just causes more and more slowing down of molecular motion and slower rates of chemical reaction until a critical temperature is reached below which there is insufficient energy for the most mobile molecules in the fluid to move appreciably over the time scale of a typical laboratory observation. At this temperature, this system almost completely loses its fluidity. Therefore, cryobiology is the branch of biology that studies life at below normal temperature. 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 So, cryobiology, once again I am repeating, cryobiology is the branch of biology that studies life at a below normal temperature. In other words, it deals with metabolic activities. And their responses in plant and animal cells stored at subnormal temperature. In other words, it deals with metabolic activities and their responses in plant and animals cells stored at subnormal temperature, usually at minus 196 centigrade, using liquefied nitrogen in presence of certain substance called cryoprotectants, such as dimethyl sulfoxide, polyvinyl pyrrolidone, polyethylene glycol, polyethylene oxide, glycerine, sucrose, proline, etc. So cryobiology has the following different dimension. It has a different dimension. Number one is cryosurgery, cryopreservation, vitrification. So number one is cryosurgery. What is cryosurgery? It comprises the use of number of techniques in which selective necrosis of tissues is achieved or hoped to be achieved through freezing at very low temperature. This low temperature is attained by using a specific solution called cryogen. According to Kulfik 1994, there are three major goals of treatment are improvement, cure and palliation. Mostly dipstick technique is used in cryosurgery. Dipstick is a cotton-tipped applicator that is dipped into a liquefied nitrogen at minus 196 centigrade and that is applied to a lesion until adequate necrosis has occurred. Open spray apparatus may also be used for the spraying cryogen on the lesion. Solidified carbon dioxide dry solid carbon dioxide, dry ice, solid carbon dioxide, dry ice, solid carbon dioxide, dry ice may also be applied directly to the skin with a mix of an octane. Such a treatment is called slush therapy. Slush therapy, slush therapy. Cryopreservation. Cryopreservation is a process of preserving and storing living system in a variable condition at very low temperature for future use. It also covers preservation by vitrification or ice-free cryopreservation. The technique was first used for preserving embryo and gametes for mouse in 1972, of a goat in 1976, and of human 1983. Okay, so this is a cryopreservation. It has been found that many seeds, plants, and lower animals can survive at freezing temperature. However, most human tissues and many animal and plant cells cannot. Particularly in case of plants, there is specific feature like large size, bigger vacuoles, and abundance of water create problem. To avoid destruction of cell, cryodestruction of cells, certain chemical substance known as cryoprotectants are used with decrease the rate of cryodestruction. The process faces certain risks like extracellular and intracellular. Extracellular or intracellular ice formation, which could also be mitigated by cryoprotectants. When tissues are cooled, slowly water comes out of cells and ice is formed in extracellular space. Too much ice in such spaces may cause damage to cell membrane due to crushing. In some cases, intracellular ice may also be formed, which could be fatal to the cell. When water comes out of cells, it may cause dehydration, causing extreme stress to the cell. Vitrification. What is vitrification? Vitrification is a process of preservation at extremely low temperature without freezing. Vitrification is a process at extremely low temperature without freezing. Vitrification involves the formation of a glossy or amorphous solid state which, unlike freezing, is not interestingly damage even to the most कॉम्प्लिकेटेड लिविंग सिस्टम. तो आपने देखा कि क्रायोबायोलॉजी का कितना यूज है. क्रायो सर्जरी उसमें है, क्रायो प्रिजर्वेशन है, विट्रिफिकेशन है. क्रायोलॉजी इज द ब्रांच ऑफ बायोलॉजी दैट स्टडीज लाइफ एट अ बिलो नॉर्मल टेम्परेचर. इन अदर वर्ड्स, इट डील्स विद द metabolic activities and the responses in plants and animal cells is stored at subnormal temperature. So thank you very much. Wish you all the best and good luck.